Welcome to the Biological Basis of Behavior series of videos. We will start with Chapter 2, Structure and Function of Neurons, and for this chapter I have five separate videos that I will present to you. Now I'm going to present these as five separate videos with questions embedded so that it's sort of an interactive process. These questions, when you answer them, will send an email to me telling me how you're doing. So you will get five separate videos if that's what I end up using it for, or one single long video that I will post on YouTube and connect to the learning management system. This long video has the benefit of allowing you to simply click on the speed and increase the speed by 2 times or 1.5 times, listening to the lecture pretty fast and getting the same amount of information but in a shorter period of time if you're, for example, reviewing the lecture. I like that capability because I've discovered that it is much easier to listen to long lectures on topics that I might have been exposed to at a faster rate, stopping when I need to, and then running it faster, getting everything in half that time. You can do that on the full-length video that will include the 5 components, and for the 5 separate videos, that one will be interactive, but unfortunately it won't allow you to speed up the video. That being said, let me tell you about the 5 topics we're going to cover in these 5 videos. The first one is the structure and function of neurons and glial cells. The second one is on membrane potentials, diffusion and electrostatic force. The third one is on action potential and rate law. The fourth one is on neurotransmission and communication between neurons. And the fifth one is about postsynaptic potentials. The overall objective for this chapter is for you to be able to identify the parts of the neurons and the overall process of communication within neurons and between neurons. So the act of action potential, excitatory postsynaptic potential, inhibitory postsynaptic potential, how these aspects of communication within neuron happen, and then how 2 neurons communicate with each other through the process of neurotransmission. I'd like you to, by the end of this class, to be able to describe the measurement of action potential and explain the dynamic equilibrium of ions that's responsible for membrane potential. I'd like you to also be able to describe the role of the ion channels in action potentials, specifically the sodium and potassium ions, and explain the all or none law and the rate law of action potentials. I'd like to have you be able to describe the structure of a synapse and the release of neurotransmitters and the activation of postsynaptic potentials and how that happens. Finally, by the end of the class, you'll be able to describe postsynaptic potentials in terms of ionic movement that cause them, the process that terminates them, either through reuptake or enzymatic breakdown, and the integration of postsynaptic potential as the process of computation, neural computation. With that being said, let's start with our first lecture, which is the structure and function of neurons and glia. Start out with the most common way of drawing a neuron is as follows, and let me use my pen for a little while. As you can see, the neuron is the entry point for a neuron, or in a way the ears, the structure that is listening out for input from other neurons is the dendrite. And there are hundreds of thousands of dendrites on any single neuron. These dendrites are in a structure sometimes called an arboreal structure, like a tree. So there is something called dendritic arborization, as in dendrites creating a tree structure. These are typically in neurons called interneurons, and we're going to talk about the different types of neurons. So dendrites, in a nutshell, are basically the listening device of the neuron. They are the input area for the neuron. They are connected to the soma, the body of the neuron. The soma is where the actual computational process and the decision making around whether to fire or not to fire an action potential takes place. In the soma also there are quite a few structures that are very important for the molecular mechanism of whether it's ion exchange, whether it's creation of receptors, whether it is the creation of neurotransmitters and then the transport of those neurotransmitters down the axon to the terminal buttons. Practically every aspect of neuronal information and is in the soma. So obviously in the soma you will find the nucleus. The nucleus is where the chromosome exist, and this is where protein production to allow for every single function of neuron takes place. So the soma, as you can tell, has a huge set of functions, but in terms of signal transmission, the soma acts as the place where the integration of signals, whether they're inhibitory or excitatory, and whether they're modulatory, this is where a lot of the integration of information takes place. At a very specific area, the junction between the soma and the axon, an area called the axon helix. You have to excuse my mouse writing. And the axon helix is where the actual final common pathway and final decision is made to trigger an action potential or not. And when you do trigger an action potential, the action potential goes down the axon. The axon is the structure that transmits the signal down to the terminal buttons. And if the dendrites are the ears, the input of a neuron, terminal buttons are the mouth of the neuron, the actual area, output area of the neuron. In the terminal buttons, you're going to find the neurotransmitter vesicles that then open up and release neurotransmitter. Between each terminal button and dendrite is an area called the synapse, the area in between the terminal button of one neuron and a dendrite of another neuron. That, in a nutshell, is the structure of a neuron in a simplistic fashion. This is the structure. It's often drawn in this way, with the cell body being more on the left and the neurotransmission going towards the right. Now, in addition to this, I need to exit that. This is an image of, once again, if you excuse me, this is an image of two neurons, or actually three neurons, communicating with each other. As you can see, this is the axon of a neuron synapsing on, so this would be maybe neuron number 1, and this is neuron number 2. Let's call this neuron number 4, and this neuron number 3. The neuron number 1 is actually, in relation to neuron number 2, is the presynaptic neuron, and a presynaptic is just, it's before the synapse. It is the neuron that is providing the terminal buttons which will release the neurotransmitter, which then will signal down to the postsynaptic neuron, neuron number 2. However, neuron number 2 itself is obviously presynaptic to neuron number 3, because it is providing the terminal buttons that then synapse on the dendrites of neuron number 3. So it's very important to keep in mind that we often call things presynaptic and postsynaptic, and that is a relative term because some presynaptic neurons are actually postsynaptic to others. So this is a very important point to keep in mind when you hear the term presynaptic and postsynaptic. Another thing in this figure that is highlighted is the myelin sheath. The myelin sheath is a covering of the axons that's very important. It has mainly two functions. One is to speed up the transmission of signals down the axon. Some axons in our bodies are several feet long. In fact, in certain animals, like a blue whale, an axon could be hundreds of feet long. So unmyelinated axons tend to be slow in their transmission. And therefore evolution has found ways to create myelinated, to create a mechanism that would speed up neural transmission. And this is the process of myelination. And we're going to talk about the details of myelination in a little while. I have to turn the pen off every now and then to move on. So now there are several types of neurons inside the nervous system, and I'm going to mention only a few right now, and as we progress in our class we're going to learn about quite a few, but the broad general types of neurons right now are multipolar neurons, and these are the most common neurons. Multipolar neurons are the most common neurons in the central nervous system. These are neurons that are extremely complex. They have one axon but many dendrites, and the axon is also very complex. It sends many terminal buttons. So the multipolar neurons tend to have extreme arborization, making it a very important neuron in terms of receiving huge amounts of information, consolidating it, making a decision whether to fire or not, and then sending information to hundreds of thousands of other neurons. One thing about multipolar neurons, as I said, is that they are most common neurons in the brain, and they're mostly in the brain, inside the brain. On the other hand, bipolar neurons and unipolar neurons are neurons that tend to be outside the brain, outside the central nervous system too, in the peripheral nervous system, and we're going to differentiate some of those. Those tend to be sensory and motor neurons, and they are relatively simple neurons in the sense that they often have a section that's basically the dendrite, a single dendrite, and here it's shown as an axon, and that's wrong from your book. It really is, this is still the dendrite, but it is acting like a single pole here, and the axon is on the other side of the cell body, and it tends to be very simple in the sense that it's a one-to-one structure. So you typically have a receptor. Receptor might respond to physical stimuli, might respond to photons, might respond to sound or movement of the cilia that is the result of a mechanical effect of sound on the fluid inside the ear. All of that sensory information then triggers a postsynaptic potential in the dendrite. And notice the word postsynaptic, so it is the postsynapse, it's the dendrite, and that then gets sent down. There is not much of a decision making. There is potentially some modulation that might say, look, don't trigger an action potential, and but there isn't much of an integration of information from multiple sources. Let's take an example of this. Imagine you're sitting here and you flick a hair on your skin, just touching it very lightly. That triggered an action potential that then reached the somatosensory cortex in your brain. This process actually was triggered by a receptor that was connected to most likely a bipolar or unipolar neuron. in which the decision was very simple, that, trigger or not trigger based on a single touch. However, obviously, your skin is not continuously feeling the stuff on it, and so it has to be inhibited some way. And so there is some modulation of these neurons, but the majority of the function is basically to trigger or not trigger. So it's relatively simple. So the fact that this neuron has two poles makes it look like a bipolar antenna, bipolar neuron, and that's what it's called, a bipolar neuron. A unipolar neuron tends to be the same structure with dendrites connected to the axon directly, with the cell body, the soma, being on the periphery, not even part of the connection. So as you can tell, the decision making is very limited here. It's just, you trigger a sensory activity and it results in a release of neurotransmitter here that then connects to another neuron that reaches the somatosensory cortex. So these are, in a nutshell, the three types of neurons we're interested in, and keep those in mind because I might be asking you questions about them at some point. Okay. This is actually a picture of a fast spiking interneuron, and you can see how arborized it is. These are all dendritic spines of the neuron, thousands and thousands of dendritic spines on these dendrites. This is the cell body, and as I say here, this is a part of the striatum, the basal ganglia, and it's a fast spiking interneuron. Again, it shows incredibly dense local axon collateral fields. So in addition to the extreme arborization, the axons themselves then split into these terminal buttons and these arbors that are very dense, allowing you to see the potential complexity of the process of decision making. This neuron is likely to receive hundreds of thousands of messages and send out hundreds of thousands of messages that are the result of a single action potential. That gives you a good example of what happens inside an interneuron, and you can think of the fact that there are billions of these neurons inside the brain, all interconnected in complex ways, talking to each other, reflecting the reality that's the outside world, and then making decisions about movement within the outside world. Now let's talk about glial cells. So we've covered the neurons, and basically we've covered the three structures we're interested in just for the sake of this chapter: the bipolar, unipolar, and interneurons. We've covered the basic structure of the neuron itself, the components, the dendrite, the soma, the axon, the terminal buttons. Now let's talk about glia, and I'm going to focus on four types of glia. The first type is astroglia, and or astrocytes, actually. I apologize. So the term is astrocytes, and astrocytes are a type of glia that have mainly four functions. One of the more important functions of astrocytes is that they connect to the capillaries in the brain, the blood vessels in the brain, in order to extract nutrition. The fact that neurons are actually very sensitive to blood, so any seepage of blood into the actual matter of the brain, what we call the parenchyma, any seepage of blood into areas where neurons exist will kill the neurons. Blood is neurotoxic. That fact has resulted in an evolutionary mechanism that protects the brain from direct contact with blood. Unlike your muscle or skin, if you prick yourself, you see blood seeping, That's because actually blood is being exchanged between the vascular system, the capillaries and the blood vessels in your body, and the muscles and the skin. Uh, and this exchange is usually through junctions, just like this, that are much bigger and open. They are open junctions that allow for exchange of blood. Blood is then blood exchanges and in fact affects those areas directly. In the brain, these junctions are closed. They're called tight junctions. They're a special type of junctions where they are actually tight. They don't allow for easy exchange of the blood. What happens at these tight junctions is that the astrocyte sort of acts like a little sucker, connecting to the tight junctions and taking the nutritional material and then transferring it, in fact, going through a cycle, a metabolic cycle, transferring what could be used by neurons to neurons. So one of the main functions of astrocyte is that they are part of what we call the blood-brain barrier. So these tight junctions and astrocyte, this composes the blood-brain barrier. That's number one. Number two, the astrocytes provide support for the whole for neurons. So in a way, this process of holding two neurons to provide the nutrition and to provide other chemicals that allow for the functioning of neurons also provides a supportive mechanism to keep neurons in place. So that's the second function. The third function is that astrocytes remove debris from the area around. So let's say there is a neuron dies for whatever reason, axons break off, and astrocytes then get rid of the debris from the area. So we've talked about several of these astrocytic functions, and I would like to just highlight where they are in your book. The astrocyte description is on page 36. We've talked about the physical support, the production of certain chemicals that help neurons. For example, astrocytes take back, they are part of the reuptake mechanism of glutamate as it breaks down. So glutamate is one of the major neurotransmitters in your brain. Astrocytes pull back the glutamine, recycle it, and create the neurotransmitter again to be sent to the neurons. So they produce some chemicals that neurons need to fulfill their function, and they are also involved in the transmission of nourishment. So these are the major functions of astrocytes. They are an extremely important glial cell in the brain. Another very important and also extremely famous glial cell is the oligodendrocyte. So the oligodendrocyte is a glial cell that inside the brain and inside actually the central nervous system acts as the source of the myelin that we talked about a few minutes ago or in the previous video. And it's basically the oligodendrocytes has processes. These are the processes that wrap around axons. And what you have to notice here is the fact that a single oligodendrocyte is wrapping around, in this case, just 3 axons. This is just an example, it could be hundreds. A single oligodendrocyte actually wraps around several axons and provide these myelin sheath, creates the myelin sheath around axons. And one thing to notice also, that the myelin sheath is not just a sheath, just a continuous sheath. In fact, in between the different myelin wrappings, are areas like this called nodes of Ranvier, and we're going to focus on them later on in this lecture. Nodes of Ranvier are nodes that allow for the regeneration of signals down an axon. So once again, a single neuron might actually be, a single axon, excuse me, might actually be wrapped up by several or multiple oligodendrocytes, and a single oligodendrocyte is likely to also wrap around multiple axons. So it's a very complex system between oligodendrocytes and axons of neurons. Now, that being said, the oligodendrocyte is actually located inside the brain and the spinal cord. On the other hand, in the peripheral nervous system, what you find is another type of cell that acts like an oligodendrocyte. This cell is called the Schwann cell. So this figure from your book shows you an oligodendrocyte and shows you how it wraps around a single, multiple axons from different neurons, and that a single oligodendrocyte might wrap around multiple axons, and a single axon will have multiple oligodendrocytes. Now, in the peripheral nervous system, it's a much simpler system. It is basically a single Schwann cell wrapping around a single axon. So it's a one-to-one relationship. This is what happens in the peripheral nervous system, and it has a very interesting clinical implication. The clinical implication is that inside the brain, recovery of axons, if there is axonal shearing or some sort of injury to an axon, the axons could technically regenerate and try to find the target they initially were connected to. Inside the brain, that's a very difficult process, since there are multiple oligodendrocytes that an axon needs to navigate. So if there is shearing of axons and you have all of these oligodendrocytes still alive and well, and an axon has to find its way back to its target, it wouldn't necessarily know which wrapping to go under. There is really, in a way, no scaffold to allow it to reach its target in an easy, direct way. Now, there is recovery in axonal injury. There's recovery in the myelin, there is recovery in the axonal injury, but it is very, very difficult in the brain to sort of recreate what you initially had. However, nerves in your body, we call neurons in the brain and bunch of axons in the brain, either tracts or fasciculi or commissures. However, in the peripheral nervous system, we call them nerves. So nerves are only in your body. Neurons and tracts and fasciculi, etc., are in your brain. So inside the body, nerves actually have a very nice scaffold ready for use if there's an injury. So if an axon gets injured, wants to find its way back home, it can use the Schwann cell to help it go back. This is why we find that nerve regeneration and nerve recovery is much easier than neuronal recovery in the brain. With that being said, that's one of the clinical implications of having Schwann cells as opposed to myelin sheath, the result of oligodendrocytes. Now, another interesting clinical fact that you should be aware of is there are two diseases famous for impacting the myelin sheath. One disease is specific to the central nervous system, and it's, as you probably can guess, multiple sclerosis. And that affects the myelination inside the central nervous system. On the other hand, because of the different type of cell that creates the myelin in the peripheral nervous system, another disease, it's an autoimmune disease, affects the PNS, the peripheral nervous system. That disease, some of you might know, if I give you a hint, swine flu and the inoculation of swine flu. It was discovered that when people were inoculated against the swine flu in many years ago in the 50s, there was a little spike of what is called a disease called Guillain-Barré, and that is a disease that makes people become paralyzed. It actually is also one of the rare effects of Zika. Zika could cause Guillain-Barré. So Guillain-Barré is the result of a reaction in the peripheral nervous system and demyelination in the peripheral nervous system, while multiple sclerosis is in the central nervous system. And the reason they're different is because we're talking about different cells that will have different proteins that the immune system will attack in different ways. So that's another implication of having these two different types of neurons. So we've talked about three types of cells, right? So we've talked about astrocytes, oligodendrocytes, and Schwann cells. And unfortunately, I don't have a picture for the fourth type of cell, but in reality, it's a very simple, very interesting cell called the microglia. So microglia are obviously the smallest glia because they're micro, and they perform the function of other cells that we have in our body called phagocytes. Phagocytes the word comes from phagia, which is swallowing, and phagocytes and the word cyte, which is cell. So these cells actually eat up other cells, invading cells like bacteria or viruses. These cells, when they encounter an invading body, will actually just wrap around it and swallow it and digest it. This process is called phagocytosis, and this process is done by a specific cell inside the brain called microglia. Okay, so these are the four types of glial cells that I would like you to be aware of, and I want you to know their functions. Let's move on and talk a little bit about here by the way, and on your slides that I've put in different places, you're going to find a lot of definitions. I've removed some of these definitions, but just to remind you, nodes of Ranvier are these naked portions that are part of the myelinated axons between adjacent oligodendrocytes, oligodendrocytes and Schwann cells. Microglia are the smallest glial cell, and they act as phagocytes, and Schwann cells, I said, are in the peripheral nervous system. The astrocytes create the blood-brain barrier, which is a semi-permeable barrier in the brain that prevents the blood from seeping into the parenchyma. There is an area, a very interesting area inside the brain called area posterma. And area posterma, it just jumped. Area posterma is a region of the medulla where the blood barrier is weak, and it evolved in order to allow organisms to detect potential poisons in the blood pretty quickly and to initiate the process of vomiting right away. You don't want to totally not be able to detect certain chemicals inside your bloodstream that could cause you to die, and therefore evolution has selected for organisms that have the ability to detect that, and that's the area posterma of the brain. Now that we've covered this section of my lecture, let's move to the next section. We've covered up to now the basic structure of neurons. We've covered the glia, so we've covered the two videos I talked about. Now we're going to cover membrane potentials, diffusion, and electrostatic forces. This is a relatively intense description of what goes on inside the neuron to create what we call the membrane potential. So bear with me, and you might not want to go at 2 times the speed in the beginning, because this is going to be a little bit heavy. Let me first talk about what a potential is. If you think back to your old physics classes, you've got two types of energy, potential and kinetic energy. Potential energy is the energy that is stored somewhere because of a differential. Kinetic energy is the energy that is taking place, that is active now. So if I hold my pen like this, it has potential energy because it's above a certain point. It's being attracted by gravitational forces towards the center of the earth. If I actually just let it go, it will fall down towards the center of the earth because it has that potential energy. Something will stop it. Obviously the earth will stop it, and it can't go all the way to the center because that energy that allows it to then dig into the center it needs much more energy, but it will fall towards the center of the earth if I leave it. And obviously if I hold it like this, I can't hold it more than an hour because I'm actually expending energy to hold it in this position. This process of expansion of energy is what creates potential energy in it. Its natural state is to actually fall. This process also is mimicked in many things, artificial as well as natural items. A battery, for example, is in a nutshell a process that holds ions in different positions against each other with a membrane in between them. And those ions really want to move in the opposite direction, but they're not allowed because of the membrane. And therefore, if you give them another route, and that's when you create a circuit, that route allows them to then jump from the negative side to the positive side. The negative side has more electrons, so the negative will, allow the electrons then to fly towards the positive side. And because there is the potential energy, and you just have to open the route for it, and it will find its way. Just like if I'm holding this here, if I just let it go, it will fall. It has potential energy. Well, inside your body, there is potential energy. And the potential energy is in the form of electrostatic and diffusion. It's the result of electrostatic and diffusion forces. So let's talk a little bit about the actual state, the natural state of the membrane and its potential. Neurons have a negative charge, a more negative charge inside compared to the outside. This is the result of a fantastic balance across a membrane. So this is the neural membrane. The neural membrane is made out of a bilipid layer that is semi-permeable. So things are going to go in and out if you let them just stick around. Okay? So this is a semi-permeable membrane in a way. It's not open, but it has channels on it that open up and close. But let's leave the channels out of the picture for now. Let's focus more on what happens inside the cell that causes it to have a potential. Well, first of all, the cell itself has organic compounds inside of it. These organic compounds are large compounds, and they tend to ionize with a negative charge that is inside. And negative charges are called anions. So the two terms you might want to remember are cations and anions. Cations, I remember it. I think the book suggests this. If you think about the cartoonish eye of a cat, it's a plus. So cation, their eyes have a plus sign, so that's how you remember. Cations are positive, anions are negative. Now, these organic anions are so large that they cannot escape the inside of the cell, making the cell more negative because they are negative. Similarly, there are chloride ions inside the cell, and they tend to also be negative, adding to the negativity of the inside. However, that's also balanced by the fact that there are potassium ions inside the cell, and these ions are just more concentrated inside. So in this image, bigger boxes like this, indicate that there is more concentration of potassium on the inside compared to the outside, and smaller, obviously smaller, is the opposite. Also, pay close attention to the arrows. The arrows mean something. Uh, so let's talk. So you've seen now potassium chloride is more concentrated on the outside than it is on the inside, and sodium is concentrated on the outside compared to the inside. Well, as I said, this is a semi-permeable membrane, and there are two pressures on these ions. One pressure is the force of diffusion. The force of diffusion you've experienced on a daily basis. It's the force that abhors vacuum. And this is in a nutshell. It's not true, but this is how I would like you to remember it. Remember, if you put a single drop of fragrance in a room, what will happen? It'll, within minutes, diffuse all over the room, and you can smell it at the extreme end. Why is there such a pressure for molecules to diffuse? Because of the fact that molecules want to be equally distributed across anything that is open. Obviously, if you put a membrane, if there's a glass door between one room and another, the molecules won't go into the other room except if you open the door. Then they will rush out, and you can smell it in the other room. Another example of the force of diffusion is when you drop a little bit of sugar in your tea. Even without stirring it, the sugar will diffuse equally in a cup of tea, and within minutes you probably can taste the sugar everywhere. You stir it because that makes it faster and diffuses faster. It allows for more interactivity with molecules, making it spread equally faster. So this is the force of diffusion, and the force of diffusion is dependent on the gradient of concentration. When there is more of an ion on one side, the force is gonna push the ion to the inside, and when there is higher on the inside, it's gonna push it to the outside. So potassium wants to go to the outside. Chloride and sodium ions want to go to the inside. Now there's another force that balances this to a certain extent, which is the electrostatic force or electrostatic pressure. The electrostatic pressure is the result of a fundamental principle in electricity and magnetism, that positive similar charges tend to repel, and opposite charges tend to attract. Positive and positive repel, negative and negative repel, positive and negative attract. So there is always an electrostatic pressure for different ions to either stay. In this case, for example, potassium is positive and the outside is positive, so the electrostatic pressure actually is to send it in. And you can see the electrostatic pressure here is represented as an arrow to the inside, while the diffusion force, because of the gradient of concentration, is gonna force the potassium to go to the outside. The opposite is true about chloride. Chloride is less concentrated on the inside, so the force of diffusion is pushing it to the inside, while the electrostatic pressure is pushing it to the outside because of the fact that there is positive on the outside and it's negative. So by the force of attraction of positive to negative, it would want to stay outside, or at least, or even leave the cell to go outside, because there is also repulsion between negative and negative. What is really interesting is what you're going to observe here is sodium has two pressures against it. So sodium here is actually wants to go into the cell because of the force of diffusion, and also wants to go into the cell because of electrostatic pressure. So both forces are working on sodium. Now, if you leave this system alone, and because of the semi-permeability of this membrane, these ions will then balance across the membrane, and you're not going to have a charge. Very soon, this battery will be depleted, if you think of it as a battery, or a capacitor if you want. it will be depleted because things will move naturally. Just like if you leave a battery for a long time, it actually will just die, because even with the best membrane, if there is any sort of permeability, it's going to cross, and it's gonna then balance out. These ions will then cancel each other out, and the charge, the potential, will go away. Now, what keeps the potential inside inside the neuron? And it's a really important question. What keeps it is a very active process called the sodium potassium pump. The sodium potassium pump is principally a pump that is continuously working to keep three sodium ions outside the cell for each two potassium ions into the cell. So it's continuously pumping sodium and potassium, and this pumping is at the rate of 3 sodium to 2 potassiums inside. 3 sodium outside to 2 potassium inside. This is an active pump. It takes a lot of energy. Part of the metabolic energy you use, because your brain actually uses 20% of the energy that your body produces, although it's much smaller than 20% of your body. Yet it uses 20%. Also, it has to continuously get that energy. You can't just, unlike other parts of your body, like your muscles or your stomach, when there is a lull in the usage of the energy, it just stops. But your brain is continuously using energy. Even when you're sleeping, there is continuous need for energy. Well, the sodium potassium pump is one depleter of the energy, and it's basically an active pump that keeps sodium out of the cell and keeps potassium inside the cell in order to keep the potential going. Okay, so fundamentally what you see here is a membrane that has a potential because of a balance that to start out with there is slight imbalance, and because of the anions and the potassium and sodium pump causing that imbalance, creating a potential. With the caveat and the important point to observe that the actual ion that really insists and has the urge to go into the cell being sodium. The sodium is one of the most important ions in terms of its role in creating action potentials and graded potentials inside the brain. Now, with that being said, let's go on and add another interesting component to the membrane. So the membrane obviously are, as I said before, they are a lipid barrier. It's semi-permeable, but there are many channels on these membranes, and these channels are specialized channels. They're basically proteins that change in shape if they are closed or open, and they allow specific ions to enter. There are potassium channels, specialized potassium channels, specialized sodium channels, specialized chloride channels, etc. That being calcium channels, so these are the big 4 ions. Now, that being said, channels differ. Some channels are activated, and this is a really important point. They're activated by a change in the membrane potential. So if the membrane potential here, as I said, usually always the inside is negative, except when you have some sort of action potential, and the outside is positive. If there is any change in the membrane potential, whether there is a flipping of the membrane potential making the outside negative and the inside positive, or even a slight potential change, some of these channels will open up. These channels are called the voltage-gated channels, channels that respond to changes in the voltage of the membrane. There are other types of channels. These are ligand-gated, meaning neurotransmitter-gated, and they have a receptor site where neurotransmitters would bind to and open them. So you've got these two types, voltage-gated and receptor or neurotransmitter-gated or ligand-gated. The final type is actually, well, not final, there are many types, but for our purposes, our third type is the voltage and ligand gated. So that type of channel only opens when you have a change in voltage and you have a neurotransmitter that's trying to open the channels. You have to have the two conditions met in order to open these channels, more complex channels, as you can tell. So with that being said, um, how how does all of that then result in what we call an action potential? So now we move to our third, uh, um, um, our fourth actually. I've covered the structure of neurons and glia, the membrane potential, and now with the third, action potentials and the rate law. So one of the things I would like to start out with is to describe what happens in the process of signaling the action potential. As I said, there are two types of channels: voltage gated and ligand gated. So in the action potential, we are going to focus specifically on the voltage gated channels. And there are two types of voltage gated channels that we're going to focus on. And these are the sodium and the potassium channels. Now, the membrane potential, as I said, is negative on the inside. So we call that a potential of, it's actually about minus 70 millivolts. Okay, remember your AA battery is 1.5 volts. It could be minus or positive. You know, everything is actually positive or negative depending where you're measuring it from, right? So we're thinking this is a minus 70, with minus being, you know, more inside, more negativity inside, more anions inside, right? So that's the resting membrane, minus 70. Now, the resting membrane fluctuates. It's not always minus 70. But there is a specific point, a specific threshold, that if you hit that threshold in terms of its fluctuation, will result in a cascade of events that opens a lot of channels. So I want you to imagine a membrane here. I'm going to draw a membrane on this side that has channels that are distant from each other. And imagine that these channels are voltage gated. Let's say and I'm simplifying everything. I'm going to talk about a single ion. Let's say let's talk about a bunch of sodium ions coming in, resulting in a bit of depolarization, meaning, you know, because the sodium is coming in, things are going from minus 70 to like minus 69 to minus 68, because you're adding positive to the inside. You're actually depolarizing. You're making it move towards zero. Okay? So one thing to remember is depolarization is equivalent to moving towards zero. Hyperpolarization is moving away from zero. Those terms are all relative, and just keep in mind, depolarization, moving towards zero, meaning losing polarization. If you use that term, that would help. Depolarizing is losing polarization. Hyperpolarizing is becoming more polarized, hyper. Well, here we go. We've got some sodium coming in. The sodium comes in. Okay, great. Sodium goes around and it dissipates. It sort of hits some other ions, maybe cancel each other out. They become a non-charged molecule, and nothing happens at this channel. Now imagine instead that these channels are much more concentrated. They're next to each other, and they're much more concentrated together. Now, what do you think would happen if, let's say, sodium comes in here, or a bunch of sodium come in here into this ion, into this channel? What will happen is the membrane potential next to this channel will change, triggering an opening of the channel next door. And this process tends to cascade because all of these channels are next to each other. Well, there are areas inside a neuron, specifically at the axon helix and in the nodes of Ranvier, where the channels are really next to each other. And any triggering that reaches a certain threshold will cascade into a process that is non-stoppable. It's an all or none. If you trigger at a very low rate, with let's say 5 ions, that might not reach the threshold. It will just die. But if you trigger it at a higher threshold, if you reach the threshold of the action potential or excitation threshold, then you trigger a process, a cascade that is non-stoppable. It's just gonna happen all the way down the axon. This is called, again, the threshold of excitation, and it happens because there is a concentration of a type of channels in a certain area, specifically the axon helix that I highlighted in an earlier segment of this video, or of this series of videos. So if you look at this, what you see is sodium channels will, when this threshold is reached, sodium channels will just begin to open up, and they will open up very quickly, and they will cascade into this process of opening up. That's the first step, is the opening up of the sodium channels, and this is number 1 here. Okay. Well, actually it's number 0 here, number 1 here. It's the opening of the sodium channels. At a certain voltage, another channel that responds at a different voltage opens up, and that's the sodium channel. That's the potassium channel. As I mentioned, sodium is primed to get into the cell. Remember the membrane potential, and remember the 2 arrows of the sodium? The 2 arrows indicated that the sodium is really, one of my students likes this word, jonesing to get into the cell. He uses it. We study drugs, so he likes the term jonesing. The sodium is jonesing to go into the cell. Now that process then triggers a cascade of sodium channels opening up, and as they open up, the membrane potential keeps increasing to the point that they trigger openings of sodium channels. Now remember the balance of potassium channels. Now remember the balance of the potassium channels. The potassium wanted to go out because of the force of diffusion. They're much more concentrated on the inside, so sodium will begin to leave the cell naturally. Now at some point, you hit a non-polarized state, and then you start polarizing in the opposite direction. Well, at a certain point, sodium channels react in a funny way. They say, look, we're done. We're going to close up, and we're going to become refractory. We're not going to respond to any impulses. Even if you try to trigger us to open, we're not going to respond. So now they've become refractory and closed. So that's the state at number 3, where they are simply closed and refractory. Now, at the same time, potassium is still leaving the cell, right? So it's still leaving. What is happening here? Well, potassium is leaving, and it's a positive ion. It's leaving the cell, therefore making the inside of the cell more and more negative. It's going to reach 0, and it's just going to keep leaving while sodium is not doing anything. It's not coming in. There's nothing to balance the potassium, and potassium keeps leaving. At some point, potassium channels close, and then sodium channels reset. They say, okay, we're ready to respond again. But still, there is a little bit of a dip. You see that dip at the bottom here that basically shows that there is a hyperpolarization for part of a millisecond, where potassium is actually, there's extra potassium outside the cell, which will diffuse away, and what you get now is back to the membrane potential of -70. So this act takes place at, as I said, the area of the axon helix, and it's a process that goes down the axon. This process is called the action potential, and it either happens or does not happen. There is no graded component here. There's no gray. It's black and white. It either happens, it's triggered, or it's not triggered. And there are a few interesting tidbits here that are relevant to psychologists. One of them is the fact that the threshold of excitation, like this threshold of excitation, is not constant. It actually could change. Medications could change it. Hormonal states could change it. So certain individuals, and in fact, the best model of this is an animal model with mice. Mice have a very interesting way of having sex. They exhibit lordosis, which is basically the mouse will arch its back. The female mouse will arch its back and allow the male mouse to penetrate her. Now, this process, called lordosis, happens only at a certain period of the animal's monthly cycle, the female animal's monthly cycle, during estrus. During that period, there is a hormonal change that affects the neurons in a specific area of the brain that makes the threshold of excitation lower, and therefore more likely to be triggered by visual stimuli, by smell actually, I take it back, by smell. So this process, as you can tell, happens only under certain conditions, in this case hormonal conditions. And however, the same processes could be happening in humans. For example, certain aspects of their physiology changes throughout their development. Their physiology changes throughout throughout the day, which makes them more or less susceptible to certain medications. And that's another clinical implication that is worth keeping in mind, is that this threshold of excitation is dependent on factors such as hormonal conditions and other conditions, including medications. Okay, that in a nutshell is what the action potential is all about. Now, the action potential travels, as I said, down the axon, and there is a really interesting process that it travels in. So because most axons, as I said, are myelinated, older, more primitive axons are unmyelinated. Imagine this is an unmyelinated axon. Here are the channels, and the channels probably are next to each other, so that triggering one will open up and allow the triggering of another, because you've changed the membrane potential, right? So this is a cascade that takes place that allows the signal of sodium, or that depolarization, to go down all the way. So you're moved to the terminal button. So what you have here are the positive is turning to negative, and the negative is turning to positive. So let's say here, where things are still in the normal state, it's positive, and inside is negative. But here, where things are opening, things are actually negative on the outside and positive on the inside, right? And so this changes as it progresses. This will become negative, and this will become positive. This will become negative, and this will become positive, due to the cascade. And sodium is just cascading down. Obviously, potassium also is opening up. So there's a lot of activity. However, this activity is actually a funny sort of activity, because the channels are mechanical. It's a mechanical activity. It's electromechanical. You've got an electrical change in the membrane, and then the protein will turn around. The protein that composes that channel will twist in a certain angle, allowing the ions to slip in. Okay. Now this act is mechanical to a certain extent. It's triggered by electrical. Now that act of mechanical movement takes some time. Unlike just pure diffusion. Diffusion tends to be faster than opening channels. Just the act of sodium swimming down comfortably in fluid is an actually faster process than sodium being entered into the cell through opening of channels. Well, that's how the actual process of speeding up the signal in myelinated axons takes place. So here it goes. It's a bit complex, so let's give it a shot, okay? So you've got the axon, you've got really concentrated channels here next to each other that open up at some point, okay, allowing sodium to come in, allowing then potassium to go out, changing the membrane. Now, what's interesting is sodium and potassium um, will start just diffusing under this area of the myelin. Now, this area of the myelin does not have channels, so no channels here, no channels. Okay, sodium doesn't enter here under the myelin. It just simply diffuses, and you can see they've drawn the arrows thicker and then thinner, because in the process of diffusion, these ions will hit other ions and they might cancel out. These ions will hit other molecules and will just be impeded in their progress down the axon. So they diffuse, but they degrade under the myelin. We call this decremental conduction, meaning that they degrade under the myelin. However, fortunately, evolution obviously favored organisms in which the myelin is just at the right spacing, where by the time the sodium reaches this point, it'll trigger another opening. It's not so weak as not to trigger an opening, but it's right at the action potential threshold that will actually trigger another opening of sodium channels and potassium channels. And this whole process of an action potential gets repeated at the node. So at each node, there is a process of re-energizing or recreating the initial action potential that happened at the axon helix. Okay, so although we call it all or none, it is still all or none. It's going to either go down or not go down, but it's going down at nodes of Ranvier. So in a way, we call this saltatory. In a way, the signal is jumping between nodes, and it's really jumping from under, right? So it's jumping like this. There's degradation, and it jumps to the next node. And at this node of Ranvier, what will happen is there is a recreation of the action potential, and it keeps jumping. So there is an action potential that's regenerated at the node of Ranvier at every point in the process of transmission of the action potential down a myelinated axon. Once again, the trick is that actual diffusion takes less time than opening all of these channels sequentially in that cascade in an unmyelinated neuron. There are unmyelinated axons. There are unmyelinated axons. They are more primitive. They evolved earlier, but they still exist in our nervous system. Okay, let me turn the pan off, move forward. Now, another question that is of significance is, well, if the signals are an all or none, meaning they are digital signals like turn on, turn off, how does all of this variety of intensity get coded? Because if you remember from if you're a vinyl collector or you're into analog versus digital, if you're a hi-fi guy or girl, the big thing is when you're looking at analog information, the information is given fully in the full spectrum, meaning you get the intensity from nothing to the maximum intensity in terms of signal. And that's represented, let's say, on a LP, if you remember LPs, that's represented by how deep a groove is. The deeper a groove on an LP, maybe the higher the amplitude or the higher the frequency, something about and the depth is continuous. So this is not just a 1 and 0, but it's actually a continuous number. So you can represent the whole set of potential intensities by picking one level the groove will go at, right? So if it's here, it might be half the intensity as if it goes all the way. Well, neurons don't allow you to have that full range because they are really on-off switches, right? At least the part of the axon. So how does intensity get represented? How do you see brighter light or weaker light? And the answer to this is that the action potential rate, how many do you have per second or millisecond? Well, second is a more appropriate time frame because it's an action potential is about 1 millisecond. How many can you have? By the way, certain action potentials, like in the ear, they require much shorter periods, and that's why they actually are a different type of action potential, depending more on potassium than sodium. But that's a little digression. But action potentials have different lengths, and most typically is the 1 millisecond length. Bottom line, you've got this the representation of intensity is the result of the actual rate of the action potentials. How many do you get per second? And that represents intensity. So we've talked about the intensity process, and we've talked about these. Here are some just reviews, a review of some of the concepts. Let me just mention a few. So ion channels, mentioned ion channels. They typically are specialized proteins that permit specific ions to enter or leave the cells. And then you've got the voltage dependent and the ligand dependent. We mentioned those. And the all or none. The all or none is the principle that once an action potential is triggered in an axon, it's propagated without any decrement. I mean, there are decrements under the myelin, but as a whole, it does not decrement. It's an all or none process. You've got the same voltage at the end as you get in the beginning. They decrement under the myelin sheet, but it's not decremental as a whole. Rate law is the principle that the variation in the intensity of a stimulus or other information being transmitted is represented by variations in the rate at which axons fire. Basically, we're talking about the frequency of firing. That's what represents intensity. And we talked about saltatory conduction, the conduction of action potentials in the myelinated, where it happens to regenerate at each node of Ranvier. And that's why it's called saltatory. It's sort of jumping, like somersaults, the same route, jumping from one node to another. And that concept comes, it's a little bit wrong because it's not really jumping, it's just being recreated because there's enough sodium to re-trigger it at each node of Ranvier. So that in a nutshell is the section on action potentials and how they work. Now we're going to move into the concept of neurotransmission. Up to now we've talked about the structure of a neuron, structure of glias, how they work, the membrane potential, because that's the building block of the action potential. Now we're going to talk about what happens, and all of that is within a single neuron. So the next thing we're going to tackle is how do neurons communicate with each other. So in the beginning we're communicating within a single neuron. Now we're going to talk about communication across neurons. And that process has several components. So the first component that is important to this is to understand the structure of the synapse and the process of the release of neurotransmitter, the activation of receptors, and the process of postsynaptic potentials and how they work, and then the termination of postsynaptic potential and the effect of postsynaptic potentials in terms of integration. This is going to be divided into two videos for the purposes of, as I said, having questions integrated into the videos. But as a whole, you can see the whole thing in a bit. Okay, so let's talk a little bit about the synapse. We've talked about presynaptic and postsynaptic, and I hope you remember that. Can you tell me what this is? This is a terminal button, and it's clearly then a presynaptic neuron here. This is the postsynaptic neuron, and this is presynaptic. So this is a very enlarged cartoonish view of that. You can see we've got the axon reaching the terminal buttons. These are microtubules. The microtubules are the structure that allows the movement of vesicles and other material up and down the axon. There is anterograde and retrograde movement of material, and this is aided by microtubule. It's actually aided by two proteins called dynesin and kinesin, and the process goes up and down. When these neurotransmitters that are transported from the soma to the terminal button reach the terminal button, they just basically sit around waiting for an action potential. An action potential reaches the terminal button and results in a cascade. The cascade makes the synaptic vesicles dock on the membrane, opening a pore, allowing the neurotransmitters to leave. So what we're going to talk about is the details of that process. But just to orient you to this is the synaptic cleft. This is the area between the two synapses. Between the synapse and the dendrite, or what we call the postsynaptic density. So on the dendrite there is a density area called the postsynaptic density. This is the synaptic cleft. Okay. This is the presynaptic membrane, the postsynaptic membrane, and what I'd like you to notice is that this is a one distance, as some synapses are so close that it's an electrical synapse. So the process of neurotransmission happens directly through electrical impulses, as opposed to the neurochemical process that we're going to be talking about. These are the terminal buttons and the synaptic vesicles that are transported from the soma. With that quick introduction to the overall structure of the synapse and what's around it, let's move to the process of communication. I'm going to skip these and talk about what happens at the level of the synapse. So, if an action potential reaches the membrane of the terminal button, obviously the membrane will depolarize, so things will flip from minus to positive and positive to negative on the outside. Well, that ends up opening a cluster of proteins in the presynaptic membrane that ends up opening calcium channels, allowing calcium to come in. The calcium then creates a cascade that makes the vesicles that contain the neurotransmitter, such as this, dock onto the membrane. Okay, and what the calcium is doing is it's helping the process of opening the fusion pores. So these are the fusion pores. These are cluster of proteins in the presynaptic membrane sitting there. These pores, when the vesicle ducts onto those pores, the fusion pores, with the help of calcium, open up. Now, this process of opening up after the docking of the synaptic vesicle and the opening up of the synaptic pores, neurotransmitter is released into the synaptic cleft, and these molecules just begin to leave the synaptic cleft and travel to the postsynaptic membrane. As you can see, what happens to the vesicle is that it becomes part of the membrane, and in fact that's not the only way that the vesicle ends up. It could end up as part of the membrane, or it could end up as follows. You have kiss and stay, which is what we just talked about, the process of uh, um, excuse me, merge and recycle is the process we were talking about, where the 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 vesicle um, merges with the membrane. The fusion pores open. The membrane then just takes the actual vesicle. The vesicle becomes part of the membrane, and then later on, buds of the membrane are pinched and recycled into the cytoplasm, recreating the vesicles. So this is called the merge and recycle process. There is the kiss and stay, where the vesicle actually does fuse, but then closes up again and stays around for the next action potential to allow it to open and release more of the neurotransmitter. And then there is the kiss and leave, where the vesicle actually releases its neurotransmitter and then leaves and gets recycled, filled up with a neurotransmitter again, and recycled through the process again. So you've got the kiss and stay, kiss and leave, and merge and recycle. These are all processes to maintain the vesicles and to reuse them, because you don't want to be transporting a lot of this up and down the axon. So it's easier to keep some of this material and recreate it inside the terminal button. Now, we've covered the process of the release of the neurotransmitter. Now, what happens on the other end, on the postsynaptic potential? On the postsynaptic neuron. And on the postsynaptic neurons, what you're going to see are, let me set the pen, excuse me. What you're going to see are channels. These channels are, again, special channels. Unlike the sodium and potassium, these are ligand gated. These are not necessarily voltage gated. Some of them are voltage and ligand gated, but we're going to talk about the ionotropic channels that are basically ligand gated. This is an example of ionotropic channel. Okay. And this channel receives a neurotransmitter at a binding site, as you can see here. When it receives the neurotransmitter at the binding site, it opens up and allows a specific ion to enter into the cell. Now, this is often a misconception. Typically, I see it in undergraduates, but sometimes people forget, and some people think that neurotransmitters actually enter the postsynaptic neuron, and they do not. They have nothing to do in terms of their entry into the postsynaptic neuron. Their main function is just as a key in a lock. They open the door and they stay outside. They get either broken down in the synaptic cleft or taken up back into the presynaptic neuron or into an astrocyte. That's another function, remember? So either they go into the presynaptic neuron or into an astrocyte, where they get recycled. So neurotransmitters are either broken down, which is the rarer form, the more primitive form, but most neurotransmitters are recycled, and they never enter the postsynaptic neuron. What enters into the postsynaptic neurons are ions, and the ions depend on the type of channel you're opening. So different neurotransmitters will open different ion channels and change the membrane potential accordingly. So let's talk about some of these receptors and their subtypes. Okay, we've talked about the ionotropic, which is the simplest type of receptor, where the actual binding site is on the channel itself and opens that channel. There is not much metabolic activity taking place here. It's a very simple process. The neurotransmitter itself changes the membrane potential. The neurotransmitter itself changes the protein structure, making it turn and twist so that it allows the ions to seep into or rush into, sometimes like sodium would rush into the cell. Well, there are other types of receptors, and they're more complex and they take more time but do more function. So ionotropic receptors tend to open up and activate or inhibit the cell. They tend to be either excitatory or inhibitory, and not much other than that. They trigger an on-off switch. On the other hand, there are receptors that are called metabotropic. Oops, excuse my horrible handwriting. The metabotropic type of channels are basically, they use energy because they are more complex. They require several steps and therefore use energy, and therefore they have a metabolic cost to them. That's why they're called metabotropic. So what happens here is a neurotransmitter actually, it binds with a receptor that's separate and apart from the channel itself. What this binding results in is an activation of a protein called a G protein. This G protein then activates, this G protein then cleaves out an alpha subunit. This alpha subunit then goes to the actual channel, and at the channel is the process of opening. So as you can see, there is the role of the G protein, which is activated by the neurotransmitter. The neurotransmitter itself is called the first messenger. The G protein is just the G protein. Alpha subunit cleaves off. It activates the receptor site, the binding site, and opening the channel. Okay, so ions now enter the channel. Another type, more complex metabotropic, involve a second messenger. I talked about the neurotransmitter as being the first messenger, but other types of systems, metabotropic receptors, actually have a second messenger involved. They go through the three steps we talked about: the G protein, the alpha subunit. The alpha subunit then connects to something. In this case, we've got the molecule of the neurotransmitter binding to the receptor. The receptor activates the G protein. The alpha subunit cleaves off and activates an enzyme. So this is the new thing. So you've got the role of an enzyme, which then creates what we call a second messenger. These are things like ionositols. These second messengers then open up the channel, but they do another thing. They go down to the nucleus of the neuron, or go up to the nucleus of the neuron, and activate a process, potentially a process that produces new proteins that might have long-term effects. See, and this is, by the way, especially calcium type or calcium type channels, where there is an activity that goes beyond just the opening and closing and triggering an on and off action potential, but something that is long-term, such as maybe memory, for example. That would be a good example of why you would have such a structure, because you're not just responding to stimulus response in the environment, but you want to somehow have a long-term learning of what has happened in the environment. And so you need to activate proteins that then will result in changes in maybe the structure of the neuron, structure of the dendrite, that then will stick around for, let's say, days, months, years, etc. These are the metabotropic receptors in a nutshell. We're going to go through some metabotropic receptors later on. But I would like to now revisit what goes on, and this is sort of related to the balance, right? We've seen something similar to this in the balance of ions. Well, the balance of ions is taking place on both presynaptic and postsynaptic, but the balance of ions is what causes the act of membrane potentials. Here, what you're seeing is the postsynaptic potentials. So now we're going to talk about postsynaptic potentials, potentials taking place at the postsynapse, typically at the dendrite and in the around the soma, the membrane of the cell body. Postsynaptic potentials are not all or none action potentials. Action potentials are all or none. They signal messages in a digital format, yes or no, activated or not activated. Now, postsynaptic potentials are more analog. They give you the full range of possibilities. And there are two types. One type of postsynaptic potential is called the excitatory postsynaptic potential, and another type is called the inhibitory. Now, if you think about it, remember sodium. Sodium rushed into the cell in order to cause an action potential, because as it rushed into the cell early on, like if you activate something that allows sodium to come in, if you reach the threshold of excitation or a threshold of the action potential, what will happen is a cascade of sodium coming into the cell takes place. Well, before you reach that threshold, there is a balance, and sodium is coming in. It's not necessarily yet reaching the threshold. It might, the sodium might be coming in somewhere in the dendrite, and the axon healing is quite a distance away. So as sodium comes in, as channels open up like this, these channels open up in the membrane at the dendrite, the postsynaptic membrane, and sodium comes in, these sodiums will result in a movement towards depolarization. But the movement isn't triggering an action potential. It's a still graded movement, right? It still isn't minus 60. It's between minus 70 and minus 60, let's say. It's excitatory because it's moving it towards an action potential. So it's exciting the cell. Hopefully it'll trigger an action potential, but it might not. If it encounters other ions that will cancel the sodium, like chloride, well, everything goes dead, right? You balance out, there is no potential, you don't trigger the cascade. Now, sodium, as I said, is excitatory, and because it's excitatory, it's basically, that it's depolarizing, getting it closer to the action potential. That's the word excitatory, is that it's exciting it, getting it closer to the action potential. On the other hand, potassium, which by its nature, remember, it had these arrows wanting to leave. Well, potassium, when you open potassium channels, what happens is potassium typically leaves the neuron. This process of leaving the neuron causes what we call inhibitory, because as you become more negative, because potassium is positive, and what you're doing here is you're making the cell more negative on the inside by having potassium leave, causing it to be more negative on the inside, more positive on the outside. Everything is relative. And therefore, you're actually causing what we call inhibitory postsynaptic potentials. These, again, are graded. They are not action potentials. They're a full range. And as they progress towards the axon helix, these potassium might hit chloride and creating potassium chloride and cancel out. So they're not necessarily gonna cause a full blast action potential. They're still graded. They're still, in a way, analog. Now, chloride, remember, it wanted to go into the cell from that figure because there is much more chloride on the outside than on the inside. Also is inhibitory because it's gonna increase the negativity on the inside because it's coming in and it's negative. So it's inhibitory. So it's inhibitory postsynaptic potentials, IPSPs, and it's hyperpolarizing, making it even harder for the cell to trigger an action potential. Because if you're at 70 and you want to go to minus 60 to trigger an action potential, well, you're now at minus 80, so you need plus 20 millivolts to get to an action potential. So you're actually telling the cell, look, please inhibit, don't go. Chloride is actually what the channels, GABA, opens. So, diazepam and all the benzos open chloride channels, GABA chloride channels that inhibit the cell, sort of calming it down. Finally, calcium tends to actually have long-term effects, but calcium itself is obviously positive and going in tends to be excitatory. Bottom line, these potentials are taking place on the soma and the dendrites, and they're building up. They're building up either to trigger an action potential at the axon helix, so this is the axon helix, okay, or inhibiting. So if you're making it more and more negative, you're inhibiting. If you're making it more and more positive, you're exciting. And the balance, the calculation is, what will the decision be? Will there be more negative or more positive? And will that happen closer to the axon helix or further away from the axon helix? So quite a bit of decision making takes place inside the soma, depending on where the signal is coming from and how strong the signal, how fast the rate of the signal. If the signal is really, really letting sodium open up and allowing sodium to come in at a very fast rate, well, that sodium, even if it's at a long distance away from the axon helix, might reach the axon helix and trigger the action potential. If it doesn't hit inhibitory postsynaptic potential, like chloride, where it cancels out with it. So you can see there is a computational process taking place, and the computation is basically the fight between the excitatory postsynaptic potentials and the inhibitory postsynaptic potentials. These are fighting against each other to build up, to trigger an action potential if there is enough excitation and less inhibition, or to inhibit the cell, to tell it not to even trigger, even when there is more sodium sent in. It's sort of inhibiting it, making it harder for it to be triggered. This process is obviously graded. It's not like an action potential where it's a yes or no. It's actually a build-up, and it's a fight between different potentials that are graded in nature. And therefore, it's an analog process, if you want to think about it that way. It's not as simple as yes or no. It's a lot of computation that's based on analog principles, based on distance from the axon helix, based on the amount, based on the rate, based on so many factors, and based on modulation too. So there are many other things modulating this process of excitation and inhibition. So let's move to the next slide. And just touch base on this process, right? So you've got the release of the neurotransmitters, and the thing is, if the neurotransmitters just sit around the synaptic cleft, they will just keep triggering the opening of channels. So you don't want that. Otherwise, you might actually have too much of these ions, especially calcium, causing something what we call excitotoxicity. And that actually is something that happens in seizure and in alcohol-related disorders, excitotoxicity. Too much of these ions going in could kill the neurons, specifically calcium. In the case of alcohol, entry of calcium into the neuron could actually kill the neuron. Well, so you need to get rid of this, of these neurotransmitters at some point. You need to somehow figure out a way to get rid of them. One way is to use these presynaptic transporters, these transporter molecules, transporter channels that take up the neurotransmitter and recycle them. So this is the process of reuptake using the transporters on the presynaptic membrane. Another way to recycle this is to actually have the astrocytes take them up and recycle them and then send them back to the neuron. So that's another way. A third way is to deactivating them. So let's say this is acetylcholine, which is often, so sometimes acetylcholine is reuptake and sometimes it's deactivated by an enzyme called acetylcholinesterase. And as you remember, anything that ends with an ase means it's an enzyme that breaks down. In this case, it breaks down acetylcholine to the acetate group plus the choline group, and it's flushed away. It diffuses into the CSF, and it's flushed away with the CSF into the body and gotten rid of. So this process of recycling is not very effective. Obviously, the process of breaking down using enzymes is not very effective. It's better to recycle, as everybody knows. So that's another component of this process of neurotransmission and what goes on at the synapse. This figure shows you what I was talking about in terms of EPSPs and IPSPs sort of working against each other. So if there is activation, and red here represents excitatory postsynaptic potentials, if there is enough activation taking place all over, especially as it progresses towards the axon helix, if there is enough of the excitation to actually trigger an action potential, you actually have that. You have a message sent down. On the other hand, even if you have enough excitation, if on the other hand another neuron is telling this postsynaptic neuron to inhibit, if it's opening chloride channels or potassium channels that both are inhibitory, IPSPs, what you're going to get is an inhibitory process that results in no triggering of the axon helix. So this is showing you that a balance like this will result of a non-triggering of the axon helix, as opposed to a clear triggering of an axon helix because of the overabundance of EPSPs.